Hello. Hey, Ruben.
Hey, hello, how are you?
Very good. Sorry, I joined the last minute. I had to switch to another account in order to join on time. I think we have 2 minutes, right? Are you
Yeah,
ready?
no worries. I'm doing well, just getting ready.
Yeah. Boston?
Yes,
How is it? How is it? How cold is it now?
it's not that cold actually. The temperature dropped a little bit, but it's not too crazy. It's like
Oh,
still 50s, 60s.
right.
Like
Yeah. So are you like setting up a lab now at
I set up a lab 5 years ago at BU.
five
So
years
before
ago?
that, yeah, so before that I was a postdoc at the Dana-Farber Cancer Center.
No, this is wonderful. I think everyone who works in spatial biology need to know Giotto, and this is why some of our members.
Yeah,
Uh,
no, it's great. Yeah.
uh, so the slides ready. You prefer the that I can ask a little bit like some small question during the talk to make like the conversation going,
Yes,
and
please.
right? Okay. Yeah. All right. So I think we have like 1 minute left for. You want to take some water, some sip of water?
Should I start sharing already or just wait a little bit?
I think let's wait a little bit. Yeah, I
All right.
think. So Anne, should we start?
Yes, we can start now. Yeah, thanks, Ruben, and thanks, Tim, for hosting the webinar today.
All
All right,
right,
thank you so much.
right. All right, let's start, right? Good morning, good afternoon, and good evening, everyone. So thank you so much for joining another episode of BioTuring's webinar, When AI Meets Biology. So it is now my great pleasure introducing Dr. Ruben Dries. He's a faculty of medicine at Boston University with appointments in hematology and oncology and computational biomedicines. Dr. Dries is one of the lead creators of Giotto Suite. I think if we work in spatial biology, we should at least know Giotto. So this is a multiscale technology-agnostic ecosystem for spatial multiomics analysis. So today's section, spatial multiomics across technologies and design. Um, Ruben will walk us through the very fast evolving landscape of spatial technologies, from single modality to multimodals, and quickly from 2D to 3D, and a lot of computational challenges that come with them. And Ruben will introduce the architecture and capabilities of Giotto Suite, highlight how it interoperates with other spatial data ecosystems, and share applications from his lab, including upcoming advances such as scalable local database framework and an innovative physics-based model that treats transcript as interacting particles of subcellular analysis. So Dr. Dries brings a unique combination of wet lab and computational expertise with training spanning from KU Leuven, Erasmus MC, Harvard, and Dana-Farber. Um, his research centers on how cells communicate within spatial microenvironments in cancer and on building the analytic tools and algorithms needed, of course, needed to decode these interactions and very complicated data type. And I work in computer science for so long, and you know, I really think spatial data will lead to a lot of new interesting problems for the computer science community to solve. So please join me welcoming Dr. Ruben Dries. And Ruben, the stage is on yours.
All right, thank you so much for the very nice introduction. So I'm going to start sharing my slides. Here we are, share. All right. Can we see the slides?
We can see the slide.
All right, thank you so much. So the title of today's talk is Spatial Multiomics Across Technology and Designs. It will focus a lot about our software, but I think I just want to show that, like it was already said in the introduction, that the spatial field is very complex, very diverse, lots of different technologies, lots of different ways to analyze data. So we need to have this kind of very flexible platform. Oh, all right, so my lab, just to give you a quick introduction, so it started my lab about 5 years ago at BU. It's all about spatial biology, but we do approach it from multiple angles. So I love technology. I love to implement new technologies, especially spatial technology. But it's always with an idea about a larger question about oncology or systems biology in a pressing situation, or in some kind of like idea that we see in the clinic that we need to improve. And so that gives us also a unique point of view in understanding what are the limitations on the software side, both in terms of like how do you represent that type of data, but also like what are the new methods that are needed to advance the field. And so it's really in that kind of like setting that we started developing the tools that we do. And so just to give a little bit of background for those that are not too familiar. So what is spatial omics biology? Obviously, this is now already a big established field, but why is it so amazing? So I think it's a combination in a very simple sense of two fields that are kind of like joined together. One hand, you have the imaging, which has been around for like ages, where you see, for example, here in H&E. On the other hand, you can now do genomics or omics on the same type of tissue. And so with that information, for example, from transcripts, proteins, lipids, anything you can think of, you can start decoding like what are the different cells, for example, are the B cells, T cells, and you know the exact location and how it kind of like. And so it's that combination that makes it really, really powerful. But there's a lot more. There's a lot more that I think is really something we should highlight. Compared to, for example, single cell, there's no dissociation bias, or there's very little. There's another bias, maybe spatial bias, but there's no dissociation bias. And that matters, for example, if we're interested in understanding the role of adipocytes, which are these kind of like holes you see here, these big blobs. These are adipocytes. You'd almost never find them in single-cell RNA-seq data sets, or very rarely, but they have a really important role in shaping some of the tumor microenvironments that we know. Oh. There's also like the requirement of tissue needs. With spatial, you can do it on even on the smallest data set that you can think of, the smallest tissue you can think of. We even did it on like this very tiny metastatic biopsy that are like a needle thick. You can also do it on any type of tissue. You can do it at FPE, which makes it immediately amenable to the clinical situation where you can potentially think about how can we use this in the clinic. And then there's many ways that you can multiplex and multimodal, and I will show some of these examples during my talk. So since spatial is such a cool technology, obviously there were lots of labs and commercial vendors that came up with their own stories or with their own technologies, their own like specific technologies that all are kind of like unique. As a result, biologists were like, hey, this is super cool. Let's think about how we could use it to understand our tumor microenvironment or our brain development, whatever you can think of. Anything that has a kind of like a solid structure would probably benefit from some type of spatial biology. That led to a really exponential increase. You can see this inflection point here around 2019 in the number of data sets that were being produced. And now it's even like it's going way up if you kind of like extrapolate this. In addition to that, once you have data, there's a lot of additional data analysis tools that are being developed, which is great. But I do think we sometimes overlook this in-between layer, which we call like software engineering, where it's really important to think about, can all of these different data analysis kind of play together, and how can we make it so that we can really ask any type of question with any type of data set that we have. So originally, I developed Giotto when I was still a postdoc in Cheng Wen's lab at the Farber. And it was really, I think, the first real spatial framework. We developed it in 2018. It was published in 2021 because it took some time. And it wasn't really simple looking back. It was just like, hey, this is single cell data with XY coordinates. I couldn't be further off the truth. It's a lot more complex than just that one. It did have all of these comprehensive analysis. And so the reason I started early before anyone else was doing it, because we had the opportunity or the fortune to work together with Dr. Long Kai, who is at Caltech and who developed SeqFish or SeqFish Plus, which is this really cool spatial multiplexing technology. So I just want to highlight a little bit why we couldn't just stick with the original Giotto, and we had to kind of like develop something from the ground up, something completely new. So first of all, there are so many new technologies, like I said before, and they're all very, very different in terms of like number of targets, resolution, the type of data, whether it's proteomics, whether it's spatial transcriptomics, whether it's sequencing based, whether it's imaging based, the cell throughput, the shapes. Nowadays, there's a lot more. You can do spatial ataxy, you can do spatial lipidomics, everything you can think of. But also the experimental design has drastically changed. Until like, I think 2-3 years ago, a lot of labs were just doing a single sample and they were super happy. They're like, hey, we can do a single sample. That's great. You get one slide from one patient. That's maybe one paper. And this is just an example here. But nowadays, it's getting a lot more complicated. And this is just a little thing that we did. Sometimes you get like one slice from one mouse lung, and this is a mouse lung with lesions that are metastatic lesions of osteosarcoma. So this is just where we inject some of these cells in the tail vein, and that kind of like goes to the mouse lung, and they develop these lesions. And so now each of these individual lesions are potentially individual tumors, and so we should also treat them as such. So we cannot just really consider that as a single sample. It's actually multiple samples embedded in a single sample. But there's a lot more complex things that I think we can do and others can do as well. You can think about a single slice, but it's very easy nowadays to serially section your, for example, we use core needle biopsies or any type of tissue and generate a 3D data set. So how do you represent that 3D data set? A single cell sample is one, but a 3D digital tissue through serial section is a whole other ball game. And you can do a lot more. Obviously, when you have spatial transcriptomics, you can use this in between sections to also do spatial proteomics, which is something we did. You can do spatial ataxy. So all of a sudden, you can generate a spatial multimodal data set. With some of the newer recent technologies like Xenium, it's very easy to multiplex a lot of tissues. So we're also seeing really big data sets with multiple samples from multiple patients or multiple mice, anything you can think of. This is just an example of something we do. We're generating really large breast cancer data sets using Xenium platform to basically see if some of the clinical assays that are being used right now, if they still hold true if you look at them from a spatial lens. And you can go one step further. A lot of people are now looking into doing tissue microarrays. And so tissue microarrays, the way it works is you can represent many, many patients all in the same block by taking these cores. And for example, what we did for pancreatic neuroendocrine tumors, we took a core from the peritumoral environment and the intratumoral environment. And so each patient is represented by these two cores. But these tissue microarrays or tumor microarrays, however you would like to call them, you can represent 50, 100, sometimes even more patients in a single spatial flow. And that makes it really powerful because now all of a sudden you should also think about, can we do some type of like statistical modeling and associate it with clinical outcomes? And so you can start building these large cohorts of patient samples and trying to see, is there any kind of like niche microenvironment related to outcomes that you can think of? As for example, what we started noticing in this case is that we saw these tertiary lymphoid structures, so these organizations where you see a lot of like amplification or proliferation of immune cells, which is usually a prognostic factor or a good prognostic factor. So having said that, that is some of what drove us to think about how can we combine all of these different assay designs and how can we make it as flexible as possible so everybody can use that in their data sets. And so we went from Giotto to Giotto Suite, and I'm going to tell you a little bit more about exactly how we implemented Giotto Suite and give you some examples about what we can do and how we are using this to build kind of like the next layer of spatial omics in a kind of like open source environment. One thing I would just like to highlight, what we always think about is when we make something, we want to make something that is usable. There's a lot of tools that are really great, probably the best in the world, but they're not very usable for a number of reasons. And I think this is very nicely laid out in this short comment from Anna Carpenter and Shantanu Singh, where they say usable tools are the last mile bridge between what computer science makes possible and what biologists are able to put to widespread use in their research. I do think that to some extent, some of that has been now alleviated with some of these data analysis agents, where it's becoming more and more easier to apply tools on your data sets, even if you're not necessarily an expert. So is there still value in generating software, what we do, for example, with Giotto Suite? And I think the answer is definitely yes. I think even more so than any time before, where I consider this more synergy rather than replacement. I think what we can do is we can create kind of like the building blocks and some of the scientific grammar where AI can play with and even extend with it. But we can do it in a sort of safe and reproducible manner, which is sometimes an issue and working or relying too much on these agents by themselves. All right, so most of the talk, I will talk about Giotto Suite. And so it's really led by two people, George in my lab and Jocelyn, a postdoc in GC's lab at Mount Sinai. And it's a multi-package ecosystem right now in R, which consists of many different things. But the whole idea is that it really caters to everyone, whether you're a user, a developer, or anyone else, you can probably use part of it or think about how you can improve it. So how did we think about how should we build the next Giotto version, Giotto Suite? We looked at biology, and the first thing we noticed is that a lot of biology doesn't necessarily happen at the single cell scale, right? Which is what we originally kind of like coming from a single cell RNA-seq field, what we always assume. There's a lot of biology happening at the individual transcripts or at organelles, and going from a single cell, you can go through a multicellular organism, even to full tissues. So we wanted a way to basically represent all types of information from all different spatial scales. We also want to make sure that we can capture any type of technology and combine any type of technology. And that technology also happens at multiple scales, depending on what kind of technology you want to multiplex with what, whether it's multiplex, whether it's a spatial array, whether it's... And so in order to do that, one of the things we did is we also have a very flexible representation of any type of output data, whether it's images, whether it's points data, whether it's intensities, shapes. And we thought about ways to make sure that we can represent your data in a kind of like multimodal way, so that it's very easy to kind of like vertical integration, thinking about, for example, connecting epigenetics, transcriptomics, or proteomics. I already showed some of the data sets that we create in the lab, and we have a lot more where we type of think about this multimodal data sets. But it's also like this is already outdated. A lot of papers are coming out where they're doing exactly this. And I think this is where the field is going, simply because like from a single tissue from 10 micron thickness, you can create one spatial transcriptomics data set. But from another adjacent section, you can do spatial ataxia. From another, you can do spatial proteomics. You can do IF. So it's very easy to multiplex all of that while still maintaining the kind of like the tissue architecture. So here are just some papers, for example, one paper, which is really nice, where they combine MERFISH data, so spatial multiplex imaging with electron microscopy. There's a 3D data, which is something that we're also working with and we can use. And for example, spatial ataxia. And all of that can be combined. You can do 3D data combined with spatial ataxia. You can do subcellular combined with like grid-based array. Anything you can think of is really possible. And the reason we do this, just to go a little bit into some of the core architecture, is that we completely separate the features and the spatial units. And the features can be literally anything you can think of, whether that's gene expression, it's proteomics, it's maybe lipidomics, anything you can think of. And you can have many features as you want. Similarly, you can have as many spatial units as you want. It doesn't need to be a cell, it can be subcellular, it can be a cell, it can be something bigger, or you can represent your data in several different ways, depending on what level you're interested in. And so then you can start aggregating it, and you can represent all of that data and merge all of that data together in any type of flexible way that you prefer to do. And I will show some of it, how it operates. I will show some examples in the next couple of slides. A little bit more on the detail level, for some of you that might be interested in, this is a little bit how we organize all the data. So we do keep the subcellular data, image data. We consider that somewhat a raw data, so we have very flexible containers to store that data. And then we have all the other data that at some point needs to be aggregated in several different ways. And whether you do that from one layer or multiple layers, everything is really possible. And then we have several slots that connect to our larger Giotto object that allow you to do all the analysis that I will show you in the next slides.
And Ruben, is there any connections between this and spatial data formats now?
The spatial data, the one that is being used by the Python environment.
Yeah, yeah.
So we're not necessarily using ZAR or spatial data per se right now. We do have an interoperability converter in between. And that is simple to say. I think we are looking into that space. But right now, there are some limitations where I think ours is a little bit more flexible. But what we hope to do in the future is kind of like either making it as simple as possible to combine the two or go from one to the other, however you want.
Right. And these are the store in the R object, right?
Yes.
The RD.
Yes, right now these are all R objects. I will show you a few slides about at the end how we're thinking about maybe making this a little bit more scalable, because there is obviously a scaling issue if you do anything in R, the same way as in Python. But yeah, that is something we are actively working on.
All right.
So talk a little bit more about these sub-object classes. In order to make it as simple as possible to work with all of these data sets, we had to kind of like make dedicated sub-object classes that could really represent any of the data. For example, you have the points, you have the Giotto points, Giotto polygons. Anything that is represented has its own dedicated object. And that allows you to really tag, for example, the spatial unit and the feature type that is associated with that specific object, and also create some provenance. So you have a history of what is happening. Just to give an example here, this is a really quick where you show these Giotto points, and you can really use these Giotto points independently. You don't necessarily have to be, that doesn't necessarily have to be part of the larger Giotto class or Giotto object if you don't want to. If you're just like, hey, I want to explore this real quickly, you can just look at the Giotto points and turn them around. By having these separate sub-objects, you can also work a little bit easier with like all type of like spatial methods, including spatial data wrangling. That's what we call it. Like making shifts, spins, anything you need to align different data sets to each other. That is just a data set alignment, but also joining spatial objects becomes really easy, whether you want to kind of like shift them all together in the same field of view, whether you want to create a Z-stack if you have 3D data, or you don't want to make any change. And then we have a very flexible way of data aggregation. So depending on the cell segmentations or any type of like other data, you can aggregate the points and intensities and create this aggregated matrix that you can then use in downstream analysis. Just to quickly show that we are always working in a technology-agnostic manner. So everything we build is always built without having one specific technology in mind, but just having the bigger picture. And here we show just some examples that I will not show for the rest of the slides. For example, with spatial deep-seq, spatial side-seq, OpenSD, slide-seq, seq-scope. Anything is really, you can represent any type of data, you can use it, and it should work without too much issue. Right, just some key examples of what we can do and what we hope everybody else can do, and why I think it matters and why it's kind of like interesting. So first, I just want to show an example for how we use our platform to do some multi-scale analysis and just kind of like thinking about what we do. If you think, for example, about Murphy's data or any spatial imaging institute-based imaging data, where you have information about the transcripts, the individual dots, you can assign them to cells, but you can identify nuclei if you have the right type of information. You can have cells, you can have a niche, you can have a domain. So everything is really tightly integrated. And sometimes it's really helpful trying to understand how do all these different analyses that you have obtained, how are they all like locally organized. And so we make it very easy to kind of like connect these different spatial scales to each other. But then you can also use that information to think about doing some very simple comparisons. For example, how is the cell and the nuclei data coming from the same spatial domains, coming from the same spatial area? How is that different? And so one thing we did is, let's say that we wanted to know which of these transcripts, you can assign them to nuclei, we can assign them to the cytoplasm and exclude them from the nuclei. What is enriched in the nucleus versus the cytoplasm? And so if you look at this, I think this is a xenium data set. in breast cancer, then what you see that is enriched in the cytoplasm is something like these ER to Golgi transport vesicles. But in the nucleus, there is the cell that is enriched to these transcripts. So that tells you something about, if you're familiar with cell trajectories or anything, that what is actively being transcribed is usually found in the nucleus. It's a little bit more complicated than that. So maybe this indicates that your cells are actually dying, that there's whatever, that they are being attacked. Or maybe this is just a signal from cancer cells that there is some cell death proteins or cell death genes, but they're actually not doing anything. Similarly, you can also do some in silico benchmarking analysis. I wouldn't even call it benchmarking. I would just think about, for example, if you do whole cell analysis versus nuclei analysis, is this a one-to-one comparison, or are you looking at two different biological phenomena? And so we can use the same tissue area, and this is what we example here. We can easily create all the whole cell. We can assign the transcript to the whole cell versus we can assign transcript to the nuclei only, and then ask the question, if we cluster them in the same way, do they have a one-to-one relationship? And as you can see, actually, when you cluster them, there's a different story between the whole cell and nuclei, indicating that what you see as a whole cell, usually use that for cell typing, versus what is present in the nuclei might be two different stories. And I think that just really shows some of the powerful things that you can now easily do.
Okay.
And again, that is just to say that doing cell and nuclear and, for example, single cell or single nuclei RNA-seq are not the same thing. They measure like two different states of a cell, and that is something that is important to keep in mind. All right, I think this is a really important thing, and this is a lot what we do in the lab, and a lot of people struggle with this, and hopefully Giotto can, for some of you, can make that easier. It's like, how, if you have spatial multimodal data, how do you decide which segmentation tool, and how do you do registration?
Right.
And to showcase that one, we highlight the 10x Genomics data set here, which is produced by 10x Genomics himself. It has a Visium data set, it has a Xenium data set, and it has IF data as well as some single cell data. And then one issue is, you can do this really with any type of data sets, how do you align all of that? And as I said previously, we have built all these like dedicated methods to flip around or kind of like rescale, flip, shift to kind of like spins, anything that is necessary, at least for an affine transformation where you're trying to align two different data sets to each other. We don't have non-affine transformations yet. We are working on something that makes that possible. But overall, I think a simple linear transformation or an affine transformation works really well for most cases. Obviously, you don't have to do all the math behind it yourself. We developed a very simple tool, a Shiny app, where you can just use your two data sets of interest, identify landmarks, some regions that you believe are almost identical. And we feel that just doing this manually usually results in the best data. There are some like automatic ways of doing this, some deep learning tools, but they are sometimes tricky to get it to work. And so using that, it's very easy to align your Visium with your Xenium and create this aligned data set. And once you have the aligned data set, there's a lot of like little things that you might be interested in. It's just to kind of like get an idea. And we did this for Xenium, Visium, and the IF data. And so one thing you might be interested in, if you have information from your IFs or immunofluorescence, you have protein data, for example, CD20 expression or CD20 protein expression, is the associated gene that encodes for this protein, MS4A1, is that spatially colocalized? Is something you would expect, but not necessarily always the case. And you can do the same for HER2 and ERB2. So with Giotto, you can actually align that, and then you can use a whole array of really well-established spatial statistics. For example, you can run the bivariate Moran's-I, which looks for the spatial associations across multiple modalities. And you can actually see how we have a better spatial association between ERBB2 and HER2, the protein and the gene, compared to the MS4A1 and CD20. You can do the same idea with Xenium and Visium if you have that data and if it's aligned. So here we can do a little trick. We can use our very flexible spatial unit development, including making all types of bins and hexagons. And we can create a pseudo Visium from the Xenium. So we can bin the Xenium along the same way as the Visium, and so you can make a direct head-to-head comparison about this is what we see in Xenium, this is what we see in Visium. Tell us about which genes are kind of like agreeing between the two technologies, and they should agree which versus which ones do not. What we see using this data set is that overall there's a positive spatial correlation between the two. It does seem to drop. And so initially we were like, hey, what is this drop? Is this maybe just because like the probes don't work really well, or maybe there's something else, is it gene specific? But what we actually noticed is that a lot of this is just correlating with expression levels. And so if you, for example, look at some of these genes like HTC that is not really correlating well, it's really because like Visium doesn't have the same sensitivity as Xenium. And so Visium is polyadenylation, or you need to kind of like pull down the transcripts, where Xenium is in situ hybridization based. So it's very two different technology, and it just shows you how much more sensitive Xenium is. Although there might also be some other reasons why some of them don't correlate. I believe that just expression levels usually explains most of it. All right. Another really big another big question we get a lot of times is, which segmentation approach should I use? And there are many different segmentation approaches. For this data set, there was an original segmentation that is provided by 10x Space Rangers, which is a little bit of a black box. I think they explain to some extent how they do it. But there's many more. There's Stardust, Cellpose, Mesmer, Baser. So the question is, which one should you use? And does it matter? And so again, our platform, since we have all these spatial units, you can do whatever, you can use multiple spatial, segmentations and combine them and basically look at how does it change the downstream results. And just to show that in a very simple way, here's the original segmentations. Here is the Baser, which is what you see here on the right. And we looked at, if we do a joint analysis, we started comparing like what is different between the original and Baser. So one thing obviously is Baser is a different way. So it's a probabilistic assignment of transcripts to cell, developed originally by the Koshenko lab here at Harvard. Baser, the cells are a lot smaller. The original, sometimes they kind of like inflate simply because how they create segmentations. But I guess the reason why it's important is, so this is a breast cancer data set. So if you now look at differences in detected cell types, combining original versus Baser, you see two cell types, for example, where you see big differences. The stromal compartment. You have a lot more stromal cells with Baser, and you have a lot more relatively, or in the original CD8 positive T cells. So in breast cancer, if there's like two really important cell types where there's a lot of research, or there are more actually, that's definitely the cancer-associated fibroblast and CD8 positive T cells, which are really important. And so having that accurate, or thinking about what is my most accurate representation, really matters if you would like to do any type of like downstream analysis and making conclusions that are based on the real data. Along the same lines, I think it's just really I like these kind of like simple analysis because it just shows you some of the limitations and how at least you can highlight these limitations. So with imaging mass cytometry, we can do something similar. So imaging mass cytometry is a spatial proteomics-based approach. So you have your proteins, they can bind to multiple genes of interest to their epitopes, they have their metal ions. You use a laser to kind of like go over the whole platform. These ions will kind of like go off, and they will be detected by the side of by 1 micron precision, sort of which proteins are detected. So obviously, if you want to do anything at the cell level, you need to make these segmentations. So we developed these segmentations. We did actually QPAT, but you can use whatever you want. And then we had a very simple idea. Hey, what if we just shrink these segmentations? And again, since we have all of that is embedded in Giotto Suite, it's very easy to just do that and create another set of segmentations. We asked the question, like, if we now overlay these intensity images and we develop two different sets, are they the same or are they different? And so here's the original, and here is the resized version. You can clearly see that these cells are just a little bit smaller. And the reason actually we made them smaller, I didn't highlight that, is with this type of data and any type of spatial proteomics data, there is this thing what people call like bleed over or contamination from neighboring cells. So we thought, like, what if we just like shrink it a little bit? Maybe that solves the whole thing. If we do a side-by-side comparison, we actually see that for most of the cells, they kind of like fall into the same cluster bucket, which we're like, hey, maybe it doesn't really matter. But then we were thinking, like, what do people usually do with this type of data if they have clinical annotation data? They actually work at the niche level where they have individual cells, source cell, and then they build their neighboring cells and they consider these niches and they see which niches are overrepresented and associated with clinical outcomes. So now we looked at these niches and we saw, so how many cells change their identity and how does that affect the niche analysis, which is all of a sudden a lot more because it can be the source cell or any of the neighboring cells. And you can actually see how many cells are affected. So if you would do a niche analysis between the original or the shrink down version, the results might drastically differ. So even though the original was not too different, the downstream analysis might be quite different. And again, I think it's really telling and something we should think about when we build a story and we have some data that we want to show and publish. Is it how robust is it, for example, to downscaling or upscaling, and what does it tell us about the data? All right. We talk most about multimodal. If you have that multimodal data, you can sometimes also have that, what people call multiomics data, more on the same tissue. Then we also have a very just flexible way of combining this information using just combining of the networks, the underlying networks in whatever dimension reduction space you would like to do this. A very popular one is weighted nearest network approach. That is something I think originally developed by the Surab people. But you can really do this whatever weights you come up with. And I think there's a lot of new tools that think about different ways to kind of like weight and how to combine these networks.
Yeah.
We show
Can
one
we
example
also-
where
Can we also take it from the spatial way upon, because we have a, if you have multiple slides, one on transcriptome, another on proteins, if we align, if the alignment goes well to the cell level, probably we can propagate the information between transcriptome and
yeah, I think that is doable. Some people might argue that that's not really multiomics data and you might be looking at something else. I would argue that, and we have these 3D data, that if you go 10 micron or 5 micron, depending on which technology, that the tissue structure and even the cells are almost identical. And oftentimes you even capture the same cell. So I think that is a valid approach. You just have to kind of like think about, am I capturing the same cell or a slightly different cell? But I think even that is something you can take into account and address. But anyway, you can kind of like do a single feature analysis, or you can combine the two, and you can think about how to interpret the results when you have more than one modality that you're using to address. Which is something we did here as a quick example in this. I think was spatial site-seq data set from 10x in the, I don't know what this is actually. All right, where are we? Okay, I think this is another, and this goes back to an earlier question, or at least it's a little bit related, is how scalable is it? And working with out-of-memory data can be a challenge. And so the way we try to address this in Giotto Suite is really using a multi-pronged approach where we have different ways of reading the data more in a chunked fashion. We are working on storing some of the data in a database. And then we have ways to do tile processing, which has been around in the spatial field for a longer time. As I said, we have this variable binning, so you can make rasters at different resolutions. And I'll show you an example of that. And then you create a count matrix, and you can do all type of like data processing and projection approaches, which I will not discuss, but it's basically you use a subset of the data and you project it to anything else.
And the goal of creating this data is for faster query or what?
Yeah, it's just basically, so there's two approaches, right? If you have unlimited amount of, well, if you have access to a supercomputer with a lot of data, or you just have enough memory, or you can just use a cloud where there's enough, then that's not, all of this is not necessarily needed. You can just use Giotto Suite as it is and just work kind of like in memory. I do think to some extent, we also try to make spatial data analysis accessible to everyone. And with everyone, I mean everyone that has a personal computer, but maybe nothing else. And so for those people, this is something where you can run any type of large scale data sets, even on a local machine. If we analyze our really large scale data sets, most of the time, we also don't necessarily use our local machines. We just use the server or whatever cloud platform we would like to use. But a lot of this is kind of like making sure that it works on a local machine. And so one simple approach is you can analyze data at multiple resolutions. So if you look at, for example, the stereoseq data, where they profile a whole mouse embryo, it's really, really big. But if you kind of like do a larger hex bin approach, then it's actually doable. You can do a smaller one, you see all these data sets. But the approach that some students in my lab took is, hey, you just use a larger approach, and then you're interested in an area, let's say this area, which we believe is the brain. You can kind of like zoom in, you can re-segment or re-tile that area in like smaller hex bins or whatever information you have. And then you can do another level, a set of analysis at a much higher resolution level. You can apply all the different clustering approaches that we have, both spatial and non-spatial. You can see those very nicely layered structures, exactly like you expected it to see. Another little cool thing to do, and I discussed this previously, since we can do this, we can easily make a pseudovisium again. So you can use that data, create a pseudovisium, since you have kind of like the coreness of how a visium spot looks like. And I think that's interesting because when you have this pseudovisium, a lot of people are also wanting to know, like, how do I do deconvolution of this pseudovisium? What is the best tool? And there are really many tools. But by doing this, you can benchmark your deconvolution data, right? Because you can use any type of tool, and we have Spatial TWS. We also have this other one that is very popular. And you can basically use whatever you want if you can connect it. You can generate the data, but you can now immediately go back to the original data, and you can kind of like, you have a ground truth that you can compare, and you have an idea like, oh, okay, maybe if I have other Visium data, then this might work better in my case than something else.
And the base data is Xenium, right?
This one is stereoseq, but you can do, I showed it previously with Xenium. This one is with stereoseq. I mean, the pseudovisium, you can create it with any type of pseudo or with any type of subcellular data, right? It's just an idea, or it just helps you to create Visium data with a ground truth if you have that information available.
Yeah.
All right. So I think I showed this multi-pronged approach, and I think one student was working, like Eddie was working on this in my lab, helping with the Giotto Suite paper. And so basically he had this idea, like, what if we can simplify this and make it even faster, make it a lot easier for people? And so this idea was like, what if we actually use these databases and don't necessarily take everything in memory, but just keep it locally on your hard drive and use some of these new embedded analytical databases that are super fast, even on your on-disk representation, so you don't necessarily have to go and read it in memory and do everything in memory. And so we started making dbVerse, which has representations for matrix data, spatial geometries, now also sequencing data, and it can really keep everything in a local or on your hard drive. It can also scale up to something if you have a bigger hard drives or you have access to something else. But then it can do all this like operations, and we're building out this kind of framework and implementing this in Giotto so that soon in the future that everybody who doesn't have access necessarily to a large supercomputer can really efficiently run this and also build new functions using these DB Matrix, DB Spatial, and some of these other tools that we're building. Along the same lines, when we use some of that ideas, we can also build interactive visualizations that are a lot faster than any of the kind of like state-of-the-art plotting tools like ggplot or plotly. And so that's also one way that we can not only do the spatial data analysis or the function development, but we also do the visualization, because like spatial data analysis is really a combination about data science and visualization, right?
Yeah.
And we see really big improvements. And I think this is really exciting. This is also exciting in the sense that this kind of like builds a layer where everybody else can build on top. And it can potentially also like be language agnostic, whether you are building something in Python or R or whatever you want. You can use the same type of like SQL type of database. All right, so very short, we have Giotto Suite. It is already installed on Terra, but you can really install on any type of like cloud software that you want. We have some Dockerized versions and everything if you're interested in that. Interactivity, really briefly, we have also some Shiny apps. Again, just if you're interested in identifying regions based on just using your eye, you can do that. We have some more advanced apps. For example, if you have 3D data, we have a way to slice the data. We actually have some really cool 3D slicing tools that not a lot of people know about. Or maybe they know there's just not a lot of people with 3D data. But with 3D data, you can all of a sudden slice your data in different directions and create in silico 2D sections of your data sets, which I think is pretty cool. And we have that available. And finally, we also can connect directly with Vitesse, which is this visualization framework from the Galenborg lab, also here at Harvard, which makes a scalable interactive visualization a little bit easier. So right now you do have to go back and forth, so it's not necessarily automatically updated. All right, then we talked a little bit about interoperability. I cannot say too much. I mean, I can just say that we have been working really hard on making Giotto as interoperable with some of the larger ecosystems that are currently available. And think about the bioconductor, the spatial experiment, I think spatial feature experiment, going back and forth between Giotto and Seurat. And this is a little bit SquidPy, the NData platform, and this is the spatial data that we talked about earlier. And so we are thinking about, do we keep this interoperability, or maybe in the future we will adopt some of these new ways of representing the data? But it does need to kind of like play well with the database approach that we currently envisioning might be really exciting in the future. What I'm actually even more excited about is how we have connected Giotto into the very large spatial ecosystem that has been around for decades. So rather than trying to reinvent the wheel every single time, we were thinking like, how can we make some of these Giotto points and Giotto poly and some of these sub-objects, how can we just make sure that they can immediately use some of these methods and functions and classes that are available, for example, in Terra, Stars, SF, everything that has been around in the space of geology, astrology for decades. And so we have built out this information. And I think, if anything, if I have seen further than others, it's by standing on the shoulders of giants, right? Where this giraffe is the our spatial ecosystem, and we are the zebra trying to like, hey, let's see how much we can use. And I just want to show a quick example. So there's many of these tools that have been vetted and implemented already for decades. And we were thinking like, how easy would it be to use some of them, and are they useful? I think the answer is obviously yes, but we want to show what we can do. And so in a matter of a week, I think we just built something based on interpolation. So hey, let's use interpolation. And the question we had was a very simple one. So a lot of people have Visium data. And so this is a Visium platform, right? You see these individual spots and you can visualize a gene. So Panther 1, this is a mouse brain slice. And you see this kind of like pattern. It looks nice, but it's not very highly resolved. So using a platform, we can use the underlying H&E data, identify nuclei. So now we have single cell information or single nuclei information. We can convert the gene expression spots to a raster, use that interpolation functions that are existing in these R ecosystems to enhance the resolution and go using this spatial interpolation technique, and then overlay that enhanced resolution on these nuclei. And so then all of a sudden you can go from something like this to something like that, where this was your original spot level visualization versus this is your nuclei visualization. So these are your nuclei, and we kind of like, it's obviously not 100% correct because it's an approximation, but this is the number of the genes that are expressed in these nuclei. So now you can do that for all genes, and you can do the same analysis. Again, all in Giotto Suite, you can do both analysis side by side. And you go from something that looks like this to something that looks like this, where we have identified the nuclei, which you see here in blue, and we have identified what is the most likely genes that are expressed in these nuclei, and you can cluster them again. And now I believe you have a more accurate representation of the actual spatial architecture, nuclei by nuclei, and you can apply some of these cell level analysis that you otherwise couldn't really do with Visium. So just to show that how easy it is to leverage some of that spatial ecosystem and apply that on your own data sets. All right, let's see. So this is just to say that if you're interested to learn more, there's a lot more to learn about Giotto Suite. I'm only barely scratching the surface. For users, there's a lot of examples. There's tutorials. For developers, there's a whole Giotto ecosystem you can learn about, including sub-websites for the individual classes. If you have a bug, then please let us know. We are a rather small team compared to some of these other larger ecosystems, but we try our best to figure it out. I just would like to say, so George and Jocelyn, they are the ones creating most of the Giotto Suite work. But in reality, it's really a whole team, and it's a whole team in GC Wen's lab, but also my whole team. I think almost everyone at some point helps with some bugs or anything else contributing to the Giotto Suite paper. So I'm not sure how much time we have right now. Oh, I just want to say one thing. If you're interested to contribute to Giotto, then please reach out. One other thing I would like to just briefly highlight that I'm also part of this Global Alliance for Spatial Technologies, Gestalt. It's a really low-level community that if you're interested, you want to learn more about spatial biology, spatial technologies, or which tools are available, I think a lot of people are on there and helping each other. It's not really, I'm not a really big key member here. It's mostly Jasmine, Johannes, and Luciano. And so with that, depending on the time, I have one other little thing I can show, or we can stop right here.
Please go ahead.
All right. So now I'm going actually in the other direction. So I showed you that we have all this data, and data analysis is as much an art as it is science. There's a lot of decisions you have to take to get insights, right? We talked about the effect you can have depending on the segmentation, how you filter normalization cluster, everything you need to have, you need to make decisions on how to move forward. And every data set is slightly different. So one student in my lab, Wendell Choi, had a really great idea, and he has a physics background, say, what if we don't have to do that? What if we can create a more holistic and fully integrated approach? One that models spatial transcript distributions more as a system of interacting particles. So this is a system that is an approach that is grounded in a physics-based theoretical model. And what it basically results in is a local correlation landscape. And so basically the approach he takes is you have all these individual transcripts. These nodes are transcripts. And he measures them as kind of like a system of interacting particles, basically creating a local correlation landscape. But there is also like a tissue gradient. And rather than doing this at the individual level, because it takes a lot of computation time, he developed a way to basically do a mean field approximation, which is pretty fast. And so the idea would be, can you do all of that? And would that be enough once you generate that data? Would that be enough to basically do every downstream analysis without necessarily have to take a lot of decisions about segmentation or clustering or normalization?
Do you need 3D or just 2D?
You can do 2D. It actually uses 3D, but in this 3D, we have shown it in actual 3D data, what we call 3D. One he calls transcripts with XYZ information already 3D. So even Xenium, Murfish, their 2D output gives you, at a transcript level, it gives you 3D transcript coordinates. The Z-axis is relatively very thin, but it does provide that. But you can do it in 2D, but you can actually scale it to whatever you want. It is scalable to any type of like 2D or 3D data set. So if you visualize that local correlation landscape, the first thing is like, okay, that looks, to some extent, that looks pretty good to the DAPI stain. But we can also quantify that. And so we quantify that by using the two Xenium data sets, a Cosmics data set, a Merscope data set, and ask like the local correlation landscape, if we define the hills are the centroids and the kind of like the valleys are the boundaries, do they correlate to the cell segmented cells that we see using the stains? So we're not using any stains for the local correlation landscape, but let's use, and then you can actually see this is the boundary 0. It really nicely correlates with where the expected boundary is for all the different data sets. So that at least indicates that our local correlation landscape agrees with some of the stains informed cell segmentation, which I think is pretty great. We do believe that it sometimes works better and sometimes it doesn't, because the stains are not always perfect. But so once you have that, all of that data, you can then assign transcripts to the cellular origin but you don't necessarily have to build a count matrix. You can actually use the context embeddings, like these kind of like vectors that he creates, to identify individual cell types directly from this local core landscape, directly from that output data. And you can clearly see this is, again, the human breast cancer data set that we used previously that is pretty good at identifying all the additional cell types that we did in a more like classical way using Giotto Suite or any type of spatial data set. You can even go further, and it's pretty good at identifying really small subtyping. For example, now here we're subtyping only the malignant cells, only the breast cancer cells, not all the other data. And you can nicely see all this like spatially informed, based on local correlation landscape, spatially informed subtypes of breast cancer, which I think is pretty impressive that you can do all of this with minimal coding and with minimal human interference.
This is impressive. This is
Yeah.
impressive.
And then last is that, okay, this is just cell typing, but can we also say something about going back about this spatial multiscales? Is that if we can use these gene vectors and kind of like discover spatial coherent functional models, but we can do it at multiple scales since we have that kind of like local correlation interactions, these local interactions and global interactions, and we can kind of like identify genes which are shared but also have different types of like patterns depending on whether they are more like locally versus more sparsely versus more everything. So that I think this is something that we're pretty excited about. It's a different approach. It does allow you to potentially integrate more data sets together without necessarily having to do them step by step. I think that's the last one.
Perfect. Thanks so much. Questions. Does Giotto support multi-sample differential analysis?
It does support multi-sample representation and identifying. What we don't really have built out completely is do the differential expression. We have differential expression analysis, but for example, if you identify modules or interaction or niches or interactions or anything spatial programs, we don't really have the statistical tools right now to do cross-sample comparisons. We are working on that because we have that in our own data sets, and we are implementing that, and hopefully we will do that in the next weeks, if not months. But we don't have that for everything yet.
Right. And the next question from the audience. When you import the polygons, do you have functions to calculate the count metrics, essentially finding which transcript belong to which polygon?
Sorry, can you say that again?
So when you have the cell segmentation as represented as a set of polygons, and you have transcript locations, do you have functions to generate the count metrics?
Yeah. So that's the whole plan, that you can overlap and you can generate the count matrix and you can do anything downstream. You can also see which transcripts, for example, do not overlap, and that's also sometimes interesting, like which transcripts do not have a cell. It's like kind of like a floating around, and is that because like there is no cell segmentation, or is that because of another reason?
Right. And taking into the scalable, is that fast or comparing to other?
Yeah, it is really fast. We started benchmarking it, and we compare it to other tools that are currently available, and think about delayed array, delayed matrix, and it is a lot faster. For example, also the spatial component is a lot faster than anything that we have currently used, including from Terra and SF, that is already highly optimized in C++, but even then, it's a lot faster. It sometimes depends on what exactly you want to do, and that is something we're trying to figure out. Should we give more exact definitions about when to use it, when not to use it? But overall, it's sometimes like orders of magnitudes faster.
Right. And Ruben, what is the scalability of Go? Can it be applied to the whole transcriptome, and how many transcripts does it support?
Scalability of which one?
Yeah, so what is the audience want to ask about whether you support the whole transcript analysis or just like a few?
Oh no, we do. We support the whole transcript analysis. Yeah.
So the question is, how about doing interactive data alignment for TMA array?
Yeah. Well, you can do it just as a normal tissue, but what we have seen sometimes is that you almost have to cut and paste the individual cores and then align them one by one, because
Yes.
sometimes there are just differences. It also depends like which section is it right adjacent, or is it sometimes like a little bit further. Then it gets harder and harder. We have a student right now who is doing this, but I think also in that approach, we kind of like subset individual cores and then do the alignment, which is a little bit of a challenge. I think there is ways to improve this in the future, but then we probably need to expand a little bit more on this, different types of like data alignments registration that is not just affine based, but also
Yeah.
allows you to more like morph some of the input data.
Yeah, and we need another question related to the alignment. How do you integrate sequential sections in one Giotto object?
Yeah, so that is you just have several sections, you align them, and that's it. And then you just have an additional metadata tag, which is that's your Z stack. And then there are already some 3D analysis implemented, I think, including the spatial expression detection. All of that is all implemented in 3D. I think we have an example where we did this in the original data set from Murfish, where they have a 3D data set on an area in the mouse brain. So if you go to the examples there on our website, you can see how it works and what you can do with it.
Right.
Again, with 3D data, for example, the alignment is sometimes a tricky issue. We have our own 3D data sets, and we are trying to see what works best. There are some tools available, but they don't always work as well as the authors claim they do, which is true for almost any tools, right? It's not just that one.
Yeah, yeah. So the field is moving very fast, and there are a lot of new problems, right? And but the time already finished, and
Yeah.
thank you so much, Ruben, for giving an excellent talk. I myself learned a lot of things from this talk, and thank you so much for.
Yeah, thank you so much for inviting me. It was great to kind of like showcase what we are doing and in which direction we're going. Thank you so much.
All right. Thank you so much, everyone.
All right, bye-bye.
Bye-bye. Thanks, Ruben.
All right, thank you so much.